Hey kiddos, it's Mr. Arnold. Let's talk a little bit about cellular respiration. This is sort of the opposite process of photosynthesis in reaction and really in what's going on overall. So in photosynthesis, if you remember, this is our basic equation. We've got carbon dioxide and water. Light is going to be the energy source, and that then is going to produce for us a glucose molecule and some oxygen. So important to us in a couple of ways because we need the glucose to eat. Okay, that's our nutrient source. And we need the oxygen to breathe. Now, what you may not have asked yourself is, why do we need the oxygen to breathe? We need the oxygen because we're going to need it in the process of cellular respiration that we're going to see now. So basically, to switch to cellular respiration, what's going on is that we're reversing this reaction. Okay, so that everything that was on the left is now on the right and vice versa. Okay, so light's out of the equation now. Okay, we don't need light to make this happen. And basically, we've got a glucose molecule and 6 oxygens are going to break down to form carbon dioxide and water. Okay, now what also is important, really important for us out of that, is that we get energy out of that. Okay, and as we'll see as we walk through the process, there's a lot of similarities with photosynthesis. It's a lot of backwards walking from the way that we originally had that. Okay, so everything that's going on here basically is going on in the mitochondria. Okay, remember mitochondria is one of the cell organelles inside of our cell. We said that it was the power source. It's where all the energy basically for the cell comes from, and the way that it gets energy for the cells is through this process of cellular respiration. Okay, so we're going to take glucose in, we're going to break it down, and we're going to produce energy, or really what we're going to produce is ATP, which is energy for the cell. Now, if you're really astute, what you probably thought to yourself is, well, aren't mitochondria only in eukaryotic cells? Prokaryotic cells don't have any organelles, so how does that work for them? Well, that is true, okay? And so what is different is that in prokaryotic cells, all the stuff that we're going to talk about here in the mitochondria occurs in the cell membrane of the prokaryotic cells, okay? So the same process is going to occur, but instead of in a mitochondria, it's going to just occur around the outside layer. So let's real quick just talk about the part. So let's real quick talk about all the parts of the mitochondria, because all these parts are going to be pretty important to us as far as where everything is occurring. So remember that mitochondria has its own DNA. That's one of the reasons we go for the endosymbiote theory, that we think that it was originally prokaryotes that basically got swallowed up and that formed eukaryotic cells. And if you look here, what that results in is you've got two membranes, okay? You've got an outer membrane, okay, kind of this orange part that's outlined in red here. And then you've got this yellow part here that is an inner membrane. And there's a lot of space in between them. We call that space the intermembrane space. And that is going to be real important for us, okay, because the last part of our cellular respiration is actually going to occur there. We've also got an important area called the matrix, which is basically the fluid that's inside of the inner membrane. Okay, and that's where all that stuff is going to occur. So that's pretty important for us. We can also see here as ATP synthase molecules just sort of embedded. They're embedded in the inner membrane, and we'll see how that works here in a second. So essentially it all comes down to this. This is the model of cellular respiration. And what there are here is there are going to be 3 parts that we're going to be concerned with. So the first of those parts is glycolysis. The second part is the Krebs cycle. And the third part is the electron transport chain. Now, a long time ago, we would have had to memorize everything and every chemical in this to go through this. And if you take biochem in college, you'll have to do that. But fortunately for us nowadays in high school biology, we just have to sort of know what the main process is and what are the main things that we're getting out of all this. So let's break it down and talk about it all step by step here. So let me erase our numbers here. So we're going to start with glycolysis. And what glycolysis is, when you see the term lysis, that means something is being broken down. So like we had lysosomes. Lysosomes break stuff down. So in glycolysis, what's being broken down is glucose. Okay, we're breaking down that glucose. So that glucose gets broken down into 2 pyruvate molecules. Now, what also is important here, and this will come back later on, is that for this, whoops, I didn't say this before, but specifically what we're talking about in this case right now, we'll get to the other in a minute, is aerobic respiration. Aerobic means with oxygen. Okay, so we've got oxygen occurring. So what's going to occur, and we'll see why that's necessary here in a second. So what happens here is that the glucose molecule is going to get broken down into 2 pyruvate molecules. Now there's a couple of important things that are going to come back to haunt us later on as far as aerobic respiration. Basically, to make this happen, we've got to take an NAD plus molecule and turn it into an NADH molecule. Okay? So all of that results in two pyruvate molecules. Now this is pretty important because all the steps that we're going to talk about here, essentially everything gets doubled up. Okay, because there's two pyruvates that come from one glucose. Now, glycolysis itself, it takes 2 ATP to get this to happen, to get the glucose to split into the two pyruvates. But each pyruvate production results in, or each production of pyruvate results in 2 ATP. So basically, we put 4 in, we get 2 out. I'm sorry, we put 2 in and we get 4 out. So that gives us a net total here at the bottom of 2 ATP that we get from glycolysis. Okay. Now, the next step, the second part here, the Krebs cycle in this area, is that we take our pyruvate molecules and we're going to add them into this little cyclical process here. So pyruvate molecules are going to go inside. Now, so glycolysis actually happens outside of the mitochondria. And so what's going to happen is these pyruvate then are going to go into the actual mitochondria and we're going into the matrix. Okay. As they go into the matrix, what happens is that they encounter this molecule and it says pyruvate dehydrogenase complex. You don't have to worry about that. Basically, pyruvate goes in and it splits off a CO2. Okay? So that CO2 gets kicked out. Now, immediately you start to see this is where our waste products are going to come from, right? This is the CO2 that we're going to eventually breathe out. And we're not breathing it out right now, but it's coming out of the mitochondria. It's going to go through the bloodstream. It's going to come back out to the lungs, and we're going to exhale that. That turns what was a pyruvate now becomes an acetyl-CoA molecule. Okay? Now, why this is important to us is this is a 2-carbon molecule now. And this 2-carbon molecule goes into the Krebs cycle and it gets joined with a 4-carbon. Okay, so the end of the Krebs cycle is a 4-carbon. It gets joined there and it forms a 6-carbon molecule. Now, you may be thinking to yourself, hey, wasn't glucose a 6-carbon molecule? And it was. But what we're actually going to form here is a molecule of citric acid. Now, why is this important? Now, it's not super important to you guys to actually have to know every bit about it, but a lot of times the Krebs cycle is called the citric acid cycle for this reason. So it's kind of important to know that citric acid is one of the things that gets formed. Now, we're not going to worry about all the other chemicals there. There's a couple other chemicals that really matter to us that we're going to care about, is that in the breaking down of this citric acid, so the citric acid is the 6-carbon thing that gets formed, that gets broken down, and when it gets broken down into 2 really important molecules for us, NADH and FADH2. We're going to call those NAD and FAD, and those 2 things, basically what their importance is they're electron carriers, okay? They're electron receivers. And so they now have some really high energy electrons. Through the breaking of this stuff down, the electrons that are now transferred to the NAD and the FAD are really high energy. And so they then are going to get kicked over to the electron transport chain, and we're going to extract that energy out of those electrons. Now, the Krebs cycle itself also results in some ATP for us. In fact, it results in 2 ATP. And so what you see so far is that we've got 2 ATP from glycolysis, 2 ATP from the Krebs cycle. Not too bad. We're getting some energy production. But what we'll see here in a second is that the real massive amount of energy production here is going to come from the electron transport chain. What you may have also noticed that we haven't used any oxygen yet, and that also is going to occur in the electron transport chain. So, what does the electron transport chain look like? Well, first off, let's jump back just a second and remember where this is occurring, okay? So this light yellow area in here that all this is happening, this is the matrix that's inside of the inner membrane of the mitochondria. That's where the Krebs cycle occurs. If you look here, this right here is where the electron transport chain is, and what you'll notice is that it's at the membrane, okay? It's in the inner membrane, so it's in between the matrix over here and the intermembrane area over here. So it's right on that line. It's on the inner membrane of the mitochondria is where this occurs. Okay? So hopefully that makes pretty good sense. So what we've got here then is the inner membrane space is up top over here, and it doesn't say it here, but I'll write it on the screen for you. This is the matrix. Okay? So this is this blue area in here inside of the mitochondria, and then the blue up here is the little inner membrane space. And where this is all happening, you see we've got a bilipid layer there, right? A membrane, that's the inner membrane, okay, of the mitochondria is where all this is occurring. So essentially what's going to happen is that we're going to come in here with NAD, with NAD, and we're going to come in here with FAD. And what happens is that they're going to give up their electrons here, and they're going to give up their electrons into this chain, and the electrons are going to travel throughout all this chain, okay, until they finally reach their end destination. Now, there's a couple of important things here. First off, you'll notice that this looks remarkably similar to the photosynthetic electron transport chain. Same sort of thing going on, except that in photosynthesis, light is coming in to add energy at each of those steps, okay? And in cellular respiration, the electrons already have the energy. They're going to give it up through the electron transport chain. And what you'll notice here at the end is that there's a molecule here, ATP synthase. It's going the opposite direction it was in photosynthesis. In photosynthesis, this was flipped over, and this end was up here, and the ATP got kicked out there as the hydrogens went through. It's going to happen the opposite way here in the cellular respiration, or in the mitochondrial electron transport chain. So as all of this is happening here, so as we produce all of this stuff, each of these little protein areas here that are actually accepting the electrons and sort of cycling them, or not cycling them through, but putting them through the chain, as all of this happens, what you'll notice here is that what we're getting on the opposite side is a bunch of H pluses, a bunch of protons. Okay, and this should look somewhat familiar. This was also happening in photosynthesis, but sort of in the opposite sort of way. So all of these H pluses are building up here. Okay, getting a bunch of protons in the inner membrane space. Okay, all of that is starting to build up. And then this electron from each of these molecules, from NADH and from FADH2, they're going to give up their electrons. It's going to go through here. Those electrons have a lot of energy. And the last step, and it's a little hard to see here since I covered over it a little bit, the last electron acceptor in the chain is an oxygen. So this is essentially why we need to breathe. I mean, this is the oxygen that we need to complete this chain so that we get enough energy to do our cellular reactions. What happens is that the oxygen is the last thing that's going to take the electrons. That oxygen then is going to get turned into, it's going to combine with some of the H pluses, and it's going to form water. Okay? And so that's our final product. So we take the oxygen in, and it kicks off the water. Now, if we go back to the very beginning here, so our products are carbon dioxide and water. Okay? And, of course, energy. And so that, where the oxygen comes in, it's the last, it accepts the last electron, and then that's going to cycle around, it's going to produce water. And so what we have here at the end is we've got a bunch of hydrogens on the outside, we've used up our oxygen, we've produced water, okay? By another thing that we'll need to know here in a second is that by the process of taking the electrons off of these, they then become NAD plus again, okay? That's a D. And they also become an FAD H plus again, okay? So we've taken the electrons away from them. That's going to be important here in a second. So oxygen becomes water. We've got a bunch of H pluses. Now, last time we had a bunch of H pluses in photosynthesis, what happened was they want to go through this ATP synthase. This is what we call, again, chemiosmosis. Bunch of H pluses out here, not very many in here. They want to get over here. The only way they can go there is to go through the ATP synthase. Remember, it's this cool little turbine-looking thing. So all of these hydrogens that are here on the outside in the inner membrane space, they're going to go through the ATP synthase. It's going to spin it around. And what we're going to get out of this is ATP. Now, we're actually going to get a lot of ATP out of this process. In fact, this is the most ATP that we're going to get out of anything. So this is a super important part of the process here. So let's look at the total that we get out of one glucose. Out of glycolysis, we get 2 ATP. Okay? From the Krebs cycle, we get another 2 ATP. And out of the electron transport chain, we actually get 32 ATP out of that if we're talking about eukaryotic cells. Okay? Now, what that total is going to total up for us is that out of the whole process of cellular respiration, we're going to get 36 ATP for eukaryotic cells. The stuff in parentheses here is for prokaryotic cells like bacteria. They have a stage where they don't need to move something, so they don't have to expend as much energy to make this process occur. So they're going to end up with 38 ATP for every time this cycle goes through. But for eukaryotic cells, so for most plants, animals, all that kind of stuff, 36 ATP is where we're going to get kicked out of it. Now that is quite a lot, and that ATP, remember, is the energy that essentially is going to drive every other reaction that goes on in our body. Anytime in a cell that something needs to happen to build something and there needs to be energy going into it to make it happen, the ATP is what goes in there, and that's what gets used to push that forward. Now, if you recall, we said that this was all aerobic respiration. Okay, so what happens when there's no oxygen? What happens when you're sprinting or when you've been working out for a long time and you're not getting as much oxygen as you might like. Sprinting is a good example because you're using up your reserves really quickly and you can't replenish them by breathing fast enough. Like if you're just doing a normal sort of jog or something, then you're usually okay. But if you're sprinting, you're using it all up in a quick burst. And so you don't have any anymore. And so your body can still do some glycolysis without the oxygen, because remember, oxygen isn't actually needed till the electron transport chain. But here's the problem. So process of glycolysis, glucose gets split into 2-pyruvate. That happens because the NAD plus becomes an NADH. That's where we get the electrons for this to happen. Okay, but if you're not getting any oxygen and you're not going through that last step of the electron transport chain, then this doesn't go back. Okay, normally in normal aerobic respiration, NADH, remember at the end, gets changed back to NAD plus as we go through the electron transport chain. But in aerobic respiration, or anaerobic respiration rather, this doesn't happen, okay? There's no cycling back there. And so what happens eventually is we reach a point where we just flat out run out of NAD+. There's no more NAD+ to happen. Now, body has to have some way to compensate for that and to make more NAD+ out of the NADH. So there are two main pathways that's going to happen. They're basically what we call types of fermentation. So the first of these is what we call lactic acid fermentation. And essentially what happens is that the pyruvate molecules, okay, what's going to happen is they're going to get turned into a different molecule, and that molecule we call is going to be called lactate or lactic acid. Okay, so that lactic acid, and what that allows is that the process of turning pyruvate into lactic acid, okay, requires that electron that drives the NADH back to become NAD plus again. Okay, so we get this cycle back, and so now we're getting continual NAD plus. Now we can keep doing this glycolysis process so that we can get that 2 ATP out. Now obviously if we're only getting 2 ATP out every time, remember it's not going to be nearly as efficient. We want the electron transport chain to happen, so we need to have that oxygen to happen. Now what also happens is that this lactic acid, it starts to build up. One of the prime examples is that it'll build up in your muscle tissue. And so when your muscles start to feel really sore and like you can't move anymore, you can't work anymore, that's because they're getting a big buildup of lactic acid in them. The only real way to get rid of that is your body's going to have some enzymes that's going to help to break it down. But basically you have to start to get oxygen back in to help break that stuff down, because otherwise you're just continually making more and more of it. This is why after a workout your muscles really hurt, or after you've been running really fast for a long time, like say in a sprint, you're really out of breath because you're trying to get that oxygen back in to basically stop this process of making so much lactic acid. That's not the only type of fermentation. The other type of fermentation is one that we're also familiar with, and that's ethanol fermentation. And so in this process what happens is that the pyruvate molecule gets broken down actually into two things, into carbon dioxide and into ethanol or alcohol. So this is how something like wine or beer is produced, okay? So we've got sugar, okay? So like say you've got, I don't know, grape juice maybe, and you throw some yeast in there, and what happens is that they'll use up all the oxygen that they have. The yeast are little microscopic organisms, and they'll use up all the oxygen that they have, but when they run out of oxygen, then they're going to start to go to anaerobic respiration, and because they got to get this NAD plus back. And so as they do that, what they're doing is they're producing carbon dioxide, and they're turning basically the sugar into ethanol, which would be alcohol. So that's why we've got these wine barrels over here. So that's essentially the process that's going on there. Again, this allows us to regenerate our NAD+, which is pretty important to us. The CO2, you can either let it out, or if you want to keep it in there, like say in the case of beer or something like that, that becomes the bubbling, and that becomes the bubbles in the carbonation in the beer. So remember, those are anaerobic respiration. That happens when you're out of oxygen. Okay? So hopefully that helps everyone with cellular respiration.